Hi, my name is Octavia Brayley and I am a Masters by Research student at the University of Bristol, where I am studying biological sciences, and I am part of the Ecology of Vision research group. My project is looking at the effects of light pollution on the circadian rhythms of intertidal organisms. This video is going to provide a brief background to my research, followed by a description of my current experiments and associated results, and what I hope to achieve in the coming months with my project. First of all, how can we define light pollution? There are two types of light pollution. The first one is astronomical light pollution, where the view of the night sky is reduced. The second type is ecological light pollution, where the natural light patterns are altered in aquatic and terrestrial environments. The quantity of light pollution has been increasing globally over the last 6 decades by about 6% each year and is widespread, affecting 80% of the surface of the Earth, with its prevalence highest in coastal areas. One of the largest impacts of light pollution on organisms is a disruption to their biological rhythms. Most organisms have a circadian clock which controls the timings of behaviour, biological and metabolic reactions, body temperature and activity. This biological clock requires a predictable change of light during a 24-hour period to maintain its functioning. Therefore, light pollution has the potential to disrupt this important system. This means that altering the natural light levels in the environment has the ability to affect numerous biological aspects of functioning in organisms, including sleep, foraging, and reproduction in animals, as well as photosynthesis in plants, to name only a few. In mammals and vertebrates, circadian rhythms are generated in the suprachiasmatic nuclei of the hypothalamus, which contain a population of heterogeneous neurons that entrain these rhythmic patterns. These circadian rhythms are regulated by the solar cycle from a direct extension from the retina called the retinohypothalamic tract, and therefore a predictable change in light and dark conditions throughout the day is essential in maintaining rhythmicity. In contrast to this vertebrate circadian mechanism, it is thought that invertebrates generate the oscillations in their optical lobes, as opposed to the suprachiasmatic nuclei. There are many studies which highlight how light pollution can alter circadian rhythms and associated behaviour in animals. I'm going to take you through a few of these now. The first one is looking at reproduction. For example, in European blackbirds, light pollution progressed the growth of their gonads, increased testosterone production, and sped up the maturation of their reproductive system by 1 month compared to birds not exposed to artificial light. Another behaviour that can be altered by light pollution is communication. This is essential for animal survival, reproduction, and just overall organism fitness. For example, fireflies use bioluminescent flashes to locate and attract mates via reactions using an enzyme substrate mechanism using luciferase and luciferin. A study found that artificial light decreases the number of flashes per minute produced by a species of firefly and reduces the activity of courtship behaviour, therefore disrupting mating and reproductive success in this species. Foraging is another behaviour which is affected by light pollution. Many marine taxa carry out diel vertical migration, which is an important function in predator avoidance. Organisms such as zooplankton migrate to the epipelagic zone at night to exploit the ample food resources, and back to the deeper mesopelagic zone during the day to avoid predation. Circadian rhythms are very important in maintaining this behaviour. A study looked at one species of zooplankton and found that the amplitude and magnitude of migration was actually reduced. This may encourage the zooplankton to confine themselves to colder waters, which could decline their growth and reproduction. About 20% of bird species migrate twice each year from cold environments to warmer areas to exploit higher resources for optimal breeding success. It is suggested that some migrating birds are subjected to light pollution and its associated effects during the most important part of their migration. Nocturnally migrating birds are attracted to artificial light at night, and therefore may alter their flight patterns towards urban environments, which may have significant consequences for their behaviour, such as diminished flight speeds, increased vocalizations, and causing them to aggregate around light sources, which can result in fatal collisions. Sleep is another process that is affected by light pollution. Sleep is important for many biological processes in birds, mammals, and invertebrates, and can be defined as stages of decreased responsiveness and mobility. For example, sleep can allow animals to overcome and recover from stresses inflicted during the day, whilst also aiding in memory and learning. Sleep is also vital for the conservation of energy and for strengthening the immune system to prevent infection by pathogens. The primary hormone in promoting sleep is melatonin, and does so by reducing core body temperature, which decreases arousal and therefore increases the likelihood of a sleeping state. Great tits exposed to artificial light during the night are active for a longer period than usual and often start singing earlier in the morning, which may have effects on energy use and territorial behaviour. They also have a weaker immune system when exposed to white light during their roosting, making them more prone to infections such as malaria, which may be due to an increased level of stress hormones. Much like animals, plants require a routine of light and dark during a 24-hour period to maintain healthy growth and to ensure key mechanisms function correctly, such as seed germination, flower development, and leaf abscission, all of which require the correct intensity, duration, and wavelength of light. Circadian rhythms in plants have been studied in Arabidopsis and are generated by clock-controlled genes with specific hormones and chemicals regulating the biological rhythms, including auxins and calcium. The presence of artificial light encourages photosynthesis to occur at times when it usually stops or slows down, which is often seen as a positive for plant growth, especially for farmers. However, high light intensities can damage the photosynthetic apparatus in plants by disrupting the rate of electron transport, oxidation and reduction, and yield in photosystem II in their apparatus. This can cause a reduction in the efficiency of photosynthesis and therefore decreases the resulting growth yield and biomass of the plants. Artificial light has also been shown to increase the abundance of photoautotrophs, such as diatoms, in freshwater sediment communities, whilst also reducing the nocturnal respiration of the existing communities in this habitat, thereby having the potential to impact the ecology and ecosystem production. As you can see, there are many potential and confirmed negative impacts of light pollution on organisms. I will now tell you about my research, including my study aims, experimental methodology, current results, and future outlook. My research has 3 aims. The first is to explore whether crabs change their behaviour and movement patterns in the presence of artificial light. The second is to predict adaptation to increasing levels of light pollution using previous data on compound eye anatomy. And the third is to suggest mitigation strategies and propose future research avenues. I would also like to have a look to see whether different colours of lights make a difference to crab behaviour, which may lead to a novel strategy to minimise light pollution on the visual systems of organisms. My study organism is a European green crab, also called the shore crab, and I collect about 15 of these each week at the beach in Clevedon, near Bristol. I then take them back to the lab, where I get things set up for data collection. I place each crab in the aquaria in an individual pot with a stone so they can be out of the water when they want to. The water temperature and salinity are both controlled throughout the experiment. The crabs are then left for 1 week and are exposed to the specific light source for that treatment. Luckily, they are quite hardy and resilient, which make them a great study organism, and they only need to be fed once every 3 days. I fix a Raspberry Pi camera to the top of the aquaria to take images of the crabs. The camera has a fisheye lens to increase the field of view to ensure no crabs are cut off from the images. It works on a time-lapse basis and takes 1 picture each minute. It is also an infrared camera, so it shows the crab movement during the day and at night. Once the images for the treatment have been downloaded, I analyse them on Python. The program looks for differences in pixels and contrasts between the images. If the pixels have changed, this means that the crab has moved. This is done for each individual crab and is then plotted on a graph to show the activity levels across time. The data output on a graph looks like this. The x-axis shows the time with 12-hour blocks labelled, from 7 at night to 7 in the morning. The peaks in the graph indicate a high activity level and corresponds to the dark pixels on the output above. My current results show that exposure to natural light is important for crabs to maintain their circadian rhythms. When exposed to natural light, there is a clear pattern to their rhythms, with activity peaking at 7 p.m. and then decreasing for the next 12 hours, before peaking again. In contrast, when the crabs are placed in complete and continuous darkness, their activity levels are much more random and less defined, showing that they need an external light source to maintain their biological clocks. In my next set of experiments, I hope to show whether exposing the crabs to artificial light that switches on and off at the same time as the sun rises and sets during the day affects their movement patterns. I will then carry out other treatments with the artificial light, including switching it on and off each hour. I will then attach coloured filters to the artificial light source to explore whether certain colours have a lesser or larger effect on the visual system and movement patterns in the crabs. Finally, I will use my data and previous data on compound eye anatomy and adaptation to make predictions about whether crabs, and therefore other intertidal organisms, will be able to adapt to changing light conditions. Complex compound eyes are found in most arthropods, such as insects and crustaceans. The eyes are often located on eye stalks, which are unique to the arthropods, allowing a large binocular spread and a greater field of view. Compound eyes are composed of ommatidia, which contain the main structures of the eye, including the lens and receptors. The specific structure of the compound eye in each organism is largely fixed in position. However, apposition and superposition eyes can temporarily adapt to light and dark conditions. Microscopy has been used in multiple studies to visualize the photomechanical changes in the eye underpinning adaptation. For example, in dark adapted eyes of Limulus, the horseshoe crab, the microvilli within the rhabdom are thinner and narrower, the area of neighbouring microvillar membranes is larger, there is a greater number of mitochondrial granules in the retinular cells, and the eccentric cell dendrite is narrower and longer than day-adapted eyes. If artificial light is applied to the eyes of the ghost crab during the night for 2 hours, the diameters of their rhabdoms decrease to the size seen in day-adapted eyes, which reduces the sensitivity of their eyes to ensure continued functioning in high light intensities. If the light phase is continued beyond sunset, however, the diameter of the rhabdoms is smaller than those during the night-adapted phase. This reduced sensitivity to the dark hours may cause problems for anti-predatory behaviour of crabs during this time if artificial lights are suddenly switched off, such as floodlights along coastlines. This is because it can take up to 15 minutes for their eyes to adapt to external light conditions. Furthermore, it has been shown that certain light intensities cause permanent damage to the retinal layer in eyes of decapods, such as the Norway lobster. The structure of the eye can also be irreversibly damaged by irradiance applied to the surface of the water, including degeneration of photoreceptors when decapod eyes are exposed to light intensities higher to what they normally encounter, in other words, above the illumination directly from the sun and moon. And this can also damage the rhabdom. Therefore, light pollution may affect decapod eye functioning and visual systems. This information, alongside my experimental data, will enable me to make predictions about the effects of continuing light pollution to the eye structure and associated adaptations of crab eyes. I hope that the results of my experiments will allow me to suggest potential mitigation strategies to minimise negative effects of light pollution on intertidal animals. Strategies could include reducing the intensity of lights along coastlines, changing the colour of the lights, or altering the frequency with which they switch on and off. Thank you very much for watching. Please do email me if you have any questions or thoughts about my research, and please follow both myself and the Ecology of Vision Research Group on Twitter to connect with us. I really look forward to hearing from you.